Hello, in this video we will discuss about the role of DNA repair pathway in cancer. When we are going to cancer, so the DNA repair pathway, which that will be adaptability to proliferate the cell during the cancer. So this is the cell which that contain chromosome and the nucleus. Inside the nucleus chromosome and cytoplasm is here, and this is the cell membrane. So this is a cell which that contain chromosome contain histone protein 60% and 40% DNA. So here I am drawing the DNA which that contain nitrogenous bases. Adenine always bind with the thymine through hydrogen binding as an anti-parallel strand, 5' to 3 and 3' to 5', and guanine with cytosine. So the DNA is here, and central dogma for the production of protein is first transcription. So the transcription means that DNA will synthesize RNA, messenger RNA through polymerase enzyme after the unwinding the helicase enzyme used. So in this way, the translation will occur through messenger RNA. This will lead to specific protein production. This specific protein is very specific for the normal function. But when the DNA will damage, will lead to damage the protein production. Our expression of gene will lead to oncogenesis. So here is the mismatch repair. So during the cancer stage, mismatch is the possibility. In this way, during the replication, mismatch occur lead to changes the gene regulation and our expression, or inhibit, which that will lead to oncogenes activation or inhibition of the oncogene regulation or oncogene inhibitor promoter. So inhibitor gene can be blocked. So on the other hand, here is you can see this is the basically this is the nuclear excision repair. This nuclear excision repair is the nuclear excision repair provide the correct information, and this is why the pyrimidine dimerization will occur due to the radiation. So during the cancer formation, this is the occurring, but when it will not repair through enzymatically, we will discuss in the next video with the whole detail. But the radiation is the important causes for the oncogenes activation due to the pyrimidine dimerization. The thymine and cytosine will interconnect rather than the hydrogen bonding. So here is the third one is the interstrand cross linked repair. If the interstrand cross linked not repair lead to oncogenesis, so in this way remember during the cancer the adaptation of the drug and the DNA repair pathway defect can be involved in drug resistance will lead to interstrand cross linked repair inability, which that will lead to cause cancer and maintenance as well as selection and subclone expansion. So the base excision repair, if the missing of the one base lead to change the polypeptide cause cancer. So polymerase enzyme work for correct it, but during the cancer stage it will not correct because the rapidly proliferation and cell division will not lead to hold the time for correction and base excision repair. So in this way the repair will not occur, will lead to more and more selection, and subclone expansion will lead to cause malignant tumor, and that will be stage 4 of the cancer, until the more and more damage the DNA due to the single mutation. So the double strand break repair is here, which when double strand break repair not stable, the genome will lead to cause tumorigenesis, so the tumor formation. So remember. So anyhow, now we will discuss about the DNA repair pathway defects. The DNA repair pathway defects can be involved in drug resistance. So the drug resistance and DNA repair pathway defect will be occur due to the can be involved in drug resistance also, but other factors also. But here we will just understand the drug resistance. So this is the tumor shrinking due to the drug providing. So the drug resistant subclone tumor with DNA repair if defect. And this is the drug resistance will proliferate, and drug treatment will lead to not effect due to the adaptation of this cell via a drug resistance gene activation. And this basically the drug resistance relapse and selection and subclone expansion, and due to the drug which that is able to tumor shrinking, but the tumor will not shrink due to the chemotherapy or other drugs. And that is why the selection and the drug resistant relapse and selection and subclone expansion will lead to malignant tumor, while here the radiation and chemotherapy will affect, but increased genomic instability generates new driver genes and creates new subclones. So the new subclone can create will lead to malignant tumor. So that is all things you should need to understand. So the role of DNA repair pathway in cancer, this is the mechanism during the cancer. So inhibition will not occur, will lead to stage 4. The expansion and the proliferation will lead to metastasis. So here is the intervention based on this insight, means target and 4 point we will discuss. The number 1 is the targeting compensating repair pathway, analysis of circulating basically the intervention based on this insight is analysis of circulating tumor DNA to reveal somatic mutation, use of chromatin modifying agents, combination of conventional and immune checkpoint blockade therapy. So this all thing is done here, and the role of DNA repair we are discussing. We was discussing about the role of DNA repair pathway during the cancer in cancer stage. So that is why the DNA repair will not occur. If the DNA will repair, so the cancer will not occur. If not, so cancer.